Hi students, this is A. Mandri Kumar Rajesh. In this video, we are going to discuss an important kingdom called fungi. Fungi can be defined as ubiquitous eukaryotic achlorophyllous heterotrophic organisms. So, it can be defined as ubiquitous eukaryotic, then achlorophyllous heterotrophic organisms. So ubiquitous means it can found everywhere or anywhere where the life is possible. Then eukaryotic means it contains well-defined nucleus, nuclear membrane, membrane-bound organelles and nucleolus. Then achlorophyllous means they lack chlorophyll. Due to the absence of chlorophyll, it has to depend upon other organism for their nutrition. That is why they are called as heterotrophic organism. That is why they are called as heterotrophic organism. Then I give you another definition which was given by Alexopoulos. So Alexopoulos defined fungi as nucleated spore-bearing achlorophyllous organisms which generally reproduce by sexually and asexually, and whose usually filaments are branched somatic structures, or typically their cell wall is made up of chitin or cellulose or by both. So Alexopoulos defined fungi as nucleated spore-bearing achlorophyllous organisms which can reproduce sexually and asexually, and its filaments branched somatic structures which was made by the cell wall, which is surrounded by the cell wall, and it is made up of chitin or cellulose or both. Then the fungi can grow in various habitats and it shows much diversity. That means it shows much variations in their structures. Then in their reproduction, then it shows variation in their physiology. Then they developed long back in geological time scale. Their existence was found from fossil records of Precambrian period. Then information from ancient literature tell us fungi were used as food by human beings. At present, fungi are used as medicines as well as food, and they are used in some other aspects. Then the study of fungi is called as mycology. The branch which deals with the study of fungi is called as mycology. Then P.A. Micali, he was an Italian botanist who was considered as founder of mycology. P.A. Micali was an Italian botanist who was considered as founder of mycology because he was the first person to give the somatic description of fungi in his book Nova Plantarum Genera in the year 1729. So P.A. Micali was considered as founder of mycology because he was the first person to give somatic description of fungi in his book Nova Plantarum Genera in 1729. Then Anton G. Berry. Is the father of modern mycology. Anton G. Berry considered as father of modern mycology. Then Alexander Fleming, who discovered penicillin in the year 1928, and he awarded Nobel Prize for the discovery of wonderful antibiotic in the 1945. So that's all about the introduction part of fungi. Then I will give you the milestones in the fungi. So please wait a minute. Let us talk about milestones in mycology. In 1729, P.A. Micali conducted spore culture experiment. I already told you who was the founder of mycology because he devoted his life for studying fungi. Then in 1767, Fontana proved that fungi could cause disease in plants. For example, the disease like red rot of sugarcane, then blast of paddy, then rust of wheat. These are all some of the diseases which are caused by fungi. So Fontana proved that fungi can cause disease in plants in the year 1767. Then in 1873, C.H. Blackley proved fungi could cause allergy in human beings. For example, Alternaria, Aspergillus, Cladosporium and Penicillium can cause allergies in human beings. So, Blackley proved fungi can cause allergy in human beings in the year 1873. Then in 1906, A.F. Blacksley reported heterothalism in fungi. Blacksley concluded zygospore formation is a sexual process. Blacksley concluded the zygospore formation is a sexual process. In homothallic species, mycelium shows bisexual condition. While in mycelium of heterothallic species, it shows plus and minus strains. While in mycelium of heterothallic species, it shows plus and minus strains. So these two strains represents two different sexes. So these two strains represent two different sexes, and that condition is called as heterothalism, and it was reported by Blacksley in the year 1906. Then in 1952, Pandakarva and Raper reported parasexual cycle. That means in some fungi, true sexual cycle is absent, which comprises nuclear fusion and meiosis. Then that fungi get benefits through a cycle called parasexual cycle. So in some fungi, true sexual cycle is absent, which comprises nuclear fusion and meiosis, and that fungi get benefits of sexuality through a cycle called parasexual cycle, and it was reported by Pantacarva and Raper. For example, Deuteromycetes is otherwise called as imperfectate fungi because it lacks sexual reproduction. So that fungi can use this parasexual cycle. So that's all about the milestones in mycology. Then I'm going to tell about the salient features of fungi. Let me explain the salient features of fungi. Majority of fungi are made up of thin filamentous branched structure called hyphae. These hyphae get interwoven to form the plant body of fungi called mycelium. Then the cell wall of fungi is made up of polysaccharide called chitin. Chitin is nothing but polymer of N-acetyl glucosamine. Then the fungi can be categorized into two types based on the presence or absence of septum. Cenocytic mycelium means that fungi do not possess cross wall. So you can find there is no cross wall or septum in that fungi, and you can find the nucleuses are free in that cytoplasm, and that condition is called as cenocytic, and that hyphae is called as, or mycelium is called as cenocytic mycelium. For example, albigo. In higher fungi, that mycelium possesses septum or cross wall, and that mycelium is called as septate mycelium. Example, fusarium. Then Plectenchyma means fungal tissue. Plectenchyma means fungal tissue, and it is divided into two types. They are prosenchyma and pseudoparenchyma. Prosenchyma means loosely arranged network of hyphae, unfused. They are not fused and parallel to one another without losing their individuality. While in the case of pseudoparenchyma, they are closely arranged network of hyphae, and they fused and have lost their individuality. That is why they are called as pseudoparenchyma. Then holocarpic. That means the entire thallus of that fungi act as reproductive structure. So the entire thallus of that fungi act as reproductive structure. While in the case of eucarpic fungi, some region of that fungi act as reproductive structure, and that remaining regions are act as vegetative structure. The remaining regions are act as vegetative structures, and that condition is called as eucarpic. Then the fungi can reproduce by asexually and sexually. If that fungi reproduce by asexually, and that condition is called as anomorph. If that fungi reproduce asexually, that condition is called as anomorph. If that fungi reproduce by sexually, that condition is called as telomorph. If that fungi reproduce by sexually, and that condition is called as telomorph. While that fungi reproduce by sexually and asexually, that condition is called holomorph. That means the fungi can reproduce by both asexually and sexually, and that condition is called as holomorph. So it can reproduce by asexually and sexually, and that condition is called as holomorph. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.